Welcome to Science and Life.
No sooner a virus enters the body, our immune system mounts an immune response to curb or to eliminate the intruder. Part of this immune process is the generation of antibodies. There are 5 major classes of antibodies in humans. For today's discussion, only 2 classes of antibodies will be relevant. They are IgM and IgG. Ig represents immunoglobulin, another common name used for antibodies. The very first class of antibodies that will be generated towards an invading microorganism, like a virus, is the IgM class. Antibodies are produced by B cells in our immune system. When a brand new virus enters our body, it will be recognized by naive B cells. These naive B cells have not exposed to any other microbe, including the one that they are going to encounter. These naive B cells can recognize any foreign molecule or microorganism that are not self. How do these naive B cells recognize a virus? On naive B cells exist structures called B cell receptors. Out of several B cell receptors, immature or undifferentiated IgM molecules also serve as B cell receptors. These IgM on B cells recognize certain specific molecules found on the surface of a virus. These molecules that are being recognized by B cell receptor IgM molecules are called antigens. On antigen molecules, only a small specific region called an epitope is being recognized by these IgM molecules. A single antibody could have several epitopes. Similarly, there could be many molecules on a viral surface that could serve as antigens. Hence, many naive B cells could recognize multiple epitopes located in different viral antigens. Once a virus is being recognized in this manner, naive B cells become activated and start dividing to generate a large number of activated B cells. A single activated B cell that recognized a unique epitope on the virus divides and produces a cell clone of itself. Likewise, another activated B cell that recognized a different epitope produces its clone of cells. This mechanism generates a very large number of activated B cells, which in turn differentiate into 2 different cell types: plasma B cells and memory B cells. Memory B cells retain information necessary for recognition of unique epitopes on this particular virus, and they stay dormant until a second infection of the same virus occurs in the future. At that stage, these memory B cells quickly spring into action to mount an immune response. The plasma B cells are responsible for producing IgM antibodies to attack the virus. These IgM producing plasma B cells reside in the spleen. IgM molecules produced and secreted into the blood by plasma cells exist as pentamers consisting of 5 antibody molecules. An antibody is a Y-shaped molecule containing 2 antigen binding sites residing at the tips of the 2 arms. A single antibody can bind to 2 antigens or 2 epitopes. Hence, an IgM pentamer with 10 binding sites can bind a virus at 10 different places or antigens. Not all classes of antibodies exist as pentamers, which is unique to the IgM class. There is a special reason for IgM antibodies to exist as pentamers. That is because, in general, IgM antibodies have weak binding affinities towards their antigens. What is the reason behind making IgM antibodies with a weak or poor binding affinity towards viral epitopes? The reason is to get this first line of defense, which is IgM antibodies, as quickly as possible to fight the virus. IgM antibodies are produced as quickly as a matter of 2 days upon the onset of symptoms. Even if they do not have a strong binding affinity per antibody molecule, IgMs get their job done by being in a pentameric form. Although the antigen binding affinity per antibody molecule is low, a pentamer has 10 binding sites, which makes the overall binding much stronger. This phenomenon is called avidity, in which the overall binding affinity or the binding strength of a molecule is enhanced by having multiple binding sites, in this case, 10 of them in an IgM pentamer. Through this network of binding to a virus, IgM antibodies are capable of attacking the virus at the very beginning of the infection. Upon the onset of symptoms in an individual, it generally takes 2 days to generate IgM antibodies. IgMs usually stay in our blood for another 14 days. A positive test result from an antibody test designed to detect IgM antibodies generated against the SARS-CoV-2 virus indicates an active ongoing infection within that person. If such a test is done before or after this time frame of 2 to 14 days after the onset of symptoms, it will not detect IgMs and the test becomes useless. However, the PCR test that detects the viral genome will be a useful test to detect the virus even before the onset of symptoms, and even some period after a patient is recovered from COVID-19. The delay of 2 days after the manifestation of symptoms to generate IgM antibodies is called the lag period. This lag period marks the time needed for the naive B cells to recognize viral antigens, to undergo several cycles of cell divisions, and to differentiate into plasma cells. What is the reason for the IgM level to go down after 2 weeks time period? By this time the second class of antibodies, IgGs, are being generated to replace IgM antibodies. By now, through the activities of both classes of antibodies, the virus load is reduced and the patient is recovering from the infection. IgG antibodies are also produced by plasma cells that initially obtained information about viral epitopes through the IgM molecules. Plasma cells edit such information to generate high affinity binding IgG antibodies. IgG antibodies exist as monomers or as single antibody molecules with 2 high affinity binding sites. As a result, the presence of low affinity IgM antibodies is not further required and their production is diminished with the arrival of high affinity IgGs. IgG producing plasma cells migrate to the bone marrow. Generally, it takes about a week of lag time after the onset of symptoms to generate IgG antibodies. During this lag period, plasma cells that used to produce IgMs undergo an editing process to improve the affinity of epitope recognition by IgG antibodies that they are going to produce. This editing process is called somatic hypermutation, which takes place at the DNA level of antibody genes. IgG antibodies stay in an individual's blood for a long period of time even after recovering from the infection. At the moment it is not known how long the IgG antibodies generated against SARS-CoV-2 virus last in the human body. In general, after a viral infection, immunity is generated in our body to face a secondary infection by the same virus. This immunity results from existing IgG producing memory cells that were generated during the previous primary infection. The secondary immune response is quick, compared to the time taken to elicit the primary immune response. This is because all necessary viral recognition information to create high affinity binding IgGs are already stored in memory B cells. These B cells quickly spring into action to combat the virus enters into our body in the second time. During the secondary immune response, the presence of low affinity IgMs is not needed since high affinity IgGs are being produced. As a result, IgM level is low. On the other hand, IgGs are generated at a higher level. After the recovery from a SARS-CoV-2 infection, the presence of IgG antibodies suggests that there could be immunity against future infection by the same virus. However, the presence of IgG antibodies does not really mean that there will be immunity. This may be why the World Health Organization warned about the lack of evidence coming from antibody testing on immunity for the coronavirus. This statement or warning must be corrected or revised. It should be stated that the current antibody tests cannot provide convincing evidence for the presence of immunity against the SARS-CoV-2 virus. Existing antibody tests currently on the market do not detect neutralizing antibodies. However, such tests are available at individual laboratory levels. What are the neutralizing antibodies? According to the existing evidence, neutralizing antibodies are those that bind to the spike, or the S protein, found on the surface of the SARS-CoV-2. This S protein binds to the ACE2 receptor found on the surface of certain human cells, and facilitates the viral entry into these cells. So, neutralizing antibodies block viral entry into host cells, thereby prevent infection. Detection of such neutralizing antibodies in an individual recovered from COVID-19 suggests that he or she has developed an immunity against the virus. Very soon there will be a test introduced to the market to detect neutralizing antibodies against SARS-CoV-2. Such a test will be an essential tool to monitor the progress of several clinical trials in search of a vaccine against this virus. In a vaccine, what we look for is the generation of neutralizing antibodies within an individual who received a candidate vaccine. At this moment most of us are interested in knowing whether a long-term immunity against a future SARS-CoV-2 infection is generated once recovered from COVID-19. There are several pieces of evidence to support the generation of immunity after SARS-CoV-2 infection. However, for how long this immunity will last in us is currently unknown, and it is premature to know. Firstly, it is generally a known fact, and also a scientifically proven concept, that our body mounts an immune response against any invading microorganism, including SARS-CoV-2. Secondly, the use of blood plasma obtained from patients who recovered from COVID-19 disease as a therapy to treat patients with ongoing SARS-CoV-2 infection and seeing favorable outcomes. For example, speedy recovery in patients received this convalescent plasma compared to those who did not. Thirdly, the presence of neutralizing antibodies over several years in patients who recovered from infections caused by other beta coronaviruses. Examples include the presence of neutralizing antibodies for 2 years in patients recovered from SARS-CoV-1 infection, and the presence of such antibodies over 3 years in individuals who recovered from MERS-CoV. Also, a relatively short immunity, from several months to a year, has been observed in individuals who were infected with coronaviruses that cause the common cold or seasonal flu. This indicates that humans are capable of generating some degree of immunity lasting for a certain time against the corona family of viruses. Fourthly, the analysis of neutralizing antibodies in 175 patients of COVID-19 with mild symptoms in China revealed that 70% of them had a relatively higher level of neutralizing antibodies. 20% had a medium level of these antibodies and 5% of patients did not have them. Since this 5% of patients also had mild symptoms, these individuals could likely have had a neutralizing antibody that is different from the one that the antibody test designed to detect. In general, the amount, or the level, of neutralizing antibodies generated within infected individuals increases with the degree of severity of the disease that they experienced. So, on that notion, will there be an immunity generated in asymptomatic individuals? It is likely that in asymptomatic carriers the probability of inducing a strong immune response may be quite low. Even the antibody levels would be pretty low in asymptomatic cases. Within an infected person, the expression of symptoms is a result of an immune response, and the generation of antibodies is part of the immune response. The lack of symptoms in asymptomatic individuals is likely due to the virus's ability to evade or deceive the host's immune system. There are several ways that viruses deceive the host's immune system to establish a kind of symbiotic relationship. These immune evasion approaches are unique to an individual virus. In the future, we will get to know the mechanisms by which SARS-CoV-2 manipulates our immune system. Fifthly, the rhesus monkey model of SARS-CoV-2 infection revealed that the monkeys who recovered from a SARS-CoV-2 infection failed to have a second infection by the same virus. Earlier, China and South Korea reported that a number of patients who recovered from COVID-19 became susceptible to reinfection based on PCR tests taken several weeks to months after their recovery. These reports challenge the general perception of the basic biology of viral infection. It is a known and accepted fact that it is highly unlikely to have a secondary infection within a very short period, several weeks to a month, after the primary infection of a virus. This observation also casts doubt on the long-term immunity against SARS-CoV-2 infection, as well as the likely success of a future vaccine. However, recent confirmatory studies on these patients revealed that it was viral fragments in their blood that PCR tests had detected earlier, and not an intact, infective virus. This result is possible as the PCR test detects 3 separate regions of the SARS-CoV-2 genome. The presence of viral fragments could easily be detected by such a PCR test, since a patient must clear billions of viruses from the body during and after the recovery from an infection. Even residual levels of an active virus could hang in the patient's blood even a month or 2 after the full recovery. This is the reason for the advice to stay self-quarantined several weeks after the recovery from COVID-19.
Hope you learned something new.